Welcome to the USDA National Clonal Germplasm Repository in Corvallis, Oregon. I am Kim Hummer, the research leader of this gene bank. In our gene bank, we have living plant collections of temperate fruit and nut crops. Today, I'd like to focus on our strawberry collection. This is our facility which houses the National Collection of Strawberries for the USDA in Corvallis, Oregon. Our gene bank was dedicated in 1981. Global strawberry production is about twice the amount of other berry crops combined. Fresh consumption of strawberries in the US lies just behind banana, apple, watermelon, and grape. Strawberries have health benefits. They contain vitamin C, folate, manganese. They are rich in phenolic compounds, including anthocyanins. They've been designated as a significant horticultural crop by the International Treaty on Plant Genetic Resources for Food and Agriculture. The genetic resources of this crop need to be conserved for humanity. From an economic standpoint, strawberries can have a high return to farmers. There's significant production in Oregon with a farm gate value of 11 to 12 million annually. The United States produces about $2.6 billion annually, and globally, strawberries are produced at $15.6 billion annually. From a botanical point of view, the genus of strawberries is Fragaria. It includes about 24 wild species that are distributed throughout the world. Each of these dots or little squares represent a locality where wild strawberries are found. Different colors represent the different species. You can see that the species are mostly distributed through the northern hemisphere, but there is one endemic in Chile, as you can see by the colored symbols. How was the modern strawberry created? The beach strawberry grows along the Pacific coast in North and South America. These plants have small red fruits, but native peoples in Chile developed a landrace of big white round strawberries before the Europeans came to the New World. Captain Amédée Fraisier saw these white strawberries growing in Chile. This is his map of Concepcion, and where the red arrow is the location where he found these white strawberries growing that the native peoples were cultivating. Fraisier brought these white strawberries back to France in 1712. He brought them to the Versailles Gardens at that time. A young botanist named Antoine Duchesne was studying wild strawberries. This is a picture of Duchesne as an old man, but at that time he was 19 years old. Duchesne made the first crosses between Chilean and other strawberry species. He crossed the Chilean strawberry with the musk strawberry of Europe, the alpine strawberry of Europe, and a recent immigrant from America, the Virginian strawberry. The Virginian strawberry produced very large fruits and the seeds were viable. Since this success, nurserymen in France, England, and elsewhere in the world began making crosses to produce new strawberry cultivars. The National Clonal Germplasm Repository in Corvallis, Oregon preserves the strawberry collection for the USDA. There are about 2,500 plants and seeds representing 24 strawberry species that have come from around the world. The collection includes hybrids, advanced breeder selections, and named cultivars. The main living collection is preserved in containers under protected environments in greenhouses and screenhouses and shade structures. One of the important aspects of our collection is to maintain the genetic identity of the strawberries. We research the descriptions of the original release notices for each cultivar. We examine the plants to make sure that the correct morphology is present in the plants that we have. Secondly, we have a new technique of identification by molecular marker fingerprints. Now we have molecular techniques that can help us confirm the identity. Each cultivar has a unique strawberry fingerprint as displayed in this image for Fairland. We can also check the paternity and the parentage analysis of each cultivar using these fingerprints. It can help us confirm the identity of the plants that we have in our collection. Finally, there are alternative conservation techniques to storing growing plants in a container in a greenhouse. These plants can be tissue cultured in small jars and stored in the refrigerator, or little growing points called meristems can be put in cryogenic preservation. We are working with the staff of the National Laboratory for Genetic Resource Preservation in Fort Collins, Colorado, to ensure long-term secure backup conservation of the strawberry germplasm.